Hello, everyone, and welcome to today's webinar, Boost Microbial Detection Sensitivity with Unbiased Human Host DNA and RNA Depletion. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Jumpcode Genomics. To learn more about our sponsor, please visit jumpcodegenomics.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Zenas George, Senior Scientist at Jumpcode Genomics. Zenas, you may now begin your presentation.
Hi, my name is Zenas George, and I am a senior scientist here at Jumpcode Genomics. I am part of the R&D team and currently leading the development of host depletion product portfolio. My major discussion today will be on how to leverage the power of Jumpcode Genomic products to boost microbial detection with unbiased human host DNA and RNA depletion. I'll be sharing with you some of the exciting results on the newly developed host depletion product and also touch on some of the other applications of Jumpcode Genomics technology. So let's take a look at today's learning objectives. At the end of this webinar, we will understand how an unbiased depletion method gives you the ability to maximize microbial genome coverage and get higher taxonomic resolution. We will also learn how to leverage CRISPR-based depletion to cut unwanted human host molecules from your next generation sequencing libraries before sequencing. And I will show us how we have applied CRISPR-based depletion to characterize microbial populations and their impact on human health. So the question we might have is, why use Jumpcode depletion kit? Well, the answer is simple, and it is just that the vast majority of molecules sequenced in many NGS applications are uninformative. These uninformative molecules are found in single-cell data, oncology sequencing data, infectious disease sequencing data, and inherited disease sequencing data as well. And when you have an NGS data set with vast amounts of uninformative data, what you are able to detect is limited, and the cost of sequencing to the depth that could increase the detection level could be unbearable. So using Jumpcode products to remove uninformative molecules before going into sequencing enhance your detection sensitivity at a lower cost. Here I am showing a generalized workflow of CRISPRclean technology, where in this case the Cas9 guide RNA complex is used in depleting the uninformative libraries, that is the purple color lines, leaving intact the informative libraries, which are the blue color lines. Our technology slots easily into various NGS workflows. And note that the depletion workflow I'm showing here is done on an already constructed libraries, but you can also apply our technology before going into library construction. Here, I'm listing out some of the CRISPRclean product portfolio, and they are, starting from the top left, we have CRISPRclean stranded total RNA prep with ribosomal RNA depletion. This product is used in removing human, mouse, and rat ribosomal RNA from RNA-seq data. We also have CRISPRclean stranded total RNA prep with ribosomal RNA depletion. This product is used in removing human, mouse, rat, and also pan-bacterial ribosomal RNA from RNA-seq data with mixed human and bacterial cells. Another product that we have is CRISPRclean single-cell RNA boost kit, which is used in removing about 50% of sequences not used in cell type identification by single-cell determination software. This product is developed to enhance determination of single-cell population in 10x Chromium library prep. And recently, we have added another very important product to the product portfolio, and that is the all-new host depletion kit for human microbiome studies. Since this is a new product, I will dive a little bit deeper into the processes behind this exciting product and the new possibilities that it affords researchers interested in unraveling the impact of microbiome on human health. Now, let's take a quick look at the human microbiome. And from head to toe, the human body harbors different species of microbes regarded as microflora. Microflora could be commensal, in which case it's neither harmful nor beneficial. It could also be a mutualistic relationship where both human and the microbes benefit from their coexistence. Under normal circumstances, the presence of microbe is not of health concern, but we now know that depending on the abundance and variety of the microbial population at a given location in or on the human body, they could be of great impact to human health. Because of this, studying the human microbiome is of importance. And to study the human microbiome, you have to collect the sample from the site of interest. And as you can see from the table here, collecting an oral sample means that the sample you are looking at has human host material at roughly between 50 to 96%. Similar trend is also observed in many other possible sites on the human body, like the skin, nasal, and vaginal samples, where human content could be greater than 95%. So, the problem is that human-derived samples like vaginal, nasal, saliva, and blood have high host background that limits detection of microbial population, especially the low biomass ones. To characterize the microbial content in microbiome samples, researchers use 16S sequencing, but this is hypothesis-driven and can only identify known microbes effectively incapable of discovering new microbes. Current solution used in removing human host DNA capitalizes on the fact that human cells are without cell wall, while on the other hand, most microbes have cell wall. So this method uses chemicals to lyse human cells, which exposes the human DNA, which is then removed using enzymes, leaving behind the tough to lyse microbial cells. The microbial cells are then lysed and the DNA purified for microbial characterization. The problem with this method is that some very important sexually transmitted microbe, such as mycoplasma, is without cell wall and will inadvertently be lysed and removed alongside human DNA. Another problem with this method is that should there be any extracellular microbial DNA in the sample, then that will be removed alongside human DNA and the microbial DNA information will be lost. This method recommends fresh sample since freeze-thaw cycles affect microbial cell integrity. We know that having fresh sample is not always possible, which makes the demand for fresh sample unrealistic. And overall, this method has been shown to bias towards gram-positive bacteria, which makes sense because the cell wall of gram-negative bacteria is weaker than that of gram-positive bacteria and could be lysed and removed alongside human content. Because of the limitation and bias introduced by current host depletion method, Jumpcode has come up with a host depletion method that is not driven by hypothesis and is without any noticeable bias in microbial content characterization. So, I will now be walking us through Jumpcode solution to removing high host content from microbiome sample. This is a single-day workflow that can easily be applied to extract the total DNA from microbiome samples. Looking at the flowchart to the left, after total DNA extraction, the DNA is quality checked and size selected for fragments that are 15 kb and above. Once that DNA is of this fragment range, the Jumpcode protocol can be started with DNA amounts of between 50 nanogram to 1,000 nanogram. The protocol starts with repairing and blocking the ends of the DNA, followed by cleanup to remove enzyme and buffers. At this stage, the DNA is ready to be treated with CRISPR-Cas9 complex with custom guide RNA. The CRISPR-Cas9 guide RNA treatment is followed by cleanup step where enzyme and buffers are also removed. At this stage, the human host DNA content have been significantly removed, and the DNA sample can be used to make NGS libraries using a library prep kit of choice. We have successfully used Illumina's DNA library prep kit for this purpose. So, with Jumpcode solution, you can achieve unbiased view of the human microbiome because our method uses specific CRISPR-mediated depletion of the human DNA. You will also achieve improved microbial genome coverage previously obscured by human DNA. You will increase detection sensitivity of low abundant microbial species. And this is a single-day workflow that can easily be applied to extracted total DNA from microbiome sample. Now, I will be showing us some of the results we have generated using our Jumpcode host depletion method to remove human DNA content from contrived sample and clinical saliva and vaginal swab samples, where we observe an improved microbial genome coverage previously obscured by human DNA. Now, let's take a look at the bar chart to the left. On the y-axis, we have percent depletion of our target site, and the x-axis represents the samples tested. Here, our target was Atrobacter luteus, which is ALU element, of which there are over 1 million copies spread throughout the human genome, and ALU element is about 300 base pairs in length. Here, we observe that we were successful in removing approximately 99% of ALU elements from both samples represented here. I will now be showing us the read reallocation metrics from our contrived sample experiment. To the left is a graph of rates aligned to human and Zymo genomes before and after depletion. Here, the y-axis is percent rates aligned to human and Zymo microbial genome in untreated and Jumpcode's CRISPRclean treated samples. The x-axis represents the samples tested. The first bar to the left is the untreated sample and has 99.25% of rates aligned to human genome, while 0.75% of rates are aligned to Zymo microbial genome. This is followed by Jumpcode's CRISPRclean treated replicate 1, which now has 88.81% of rates aligned to human genome, while 11.19% of rates are aligned to Zymo microbial genome. The third bar graph starting from the left is replicate number 2 of Jumpcode's CRISPRclean treated sample, which now has 88.48% of rates aligned to human genome, while 11.52% of rates are aligned to Zymo microbial genome. To the right is bar chart showing how the rates aligned to the 10 Zymo microbial genomes are redistributed to each species within the community in replicate number 1. As can be seen, there is greater than 13-fold increase in rates aligned to each species, and this increase is without any obvious bias, even though this community comprises of species with differences in GC content, genome size, and gram stain status. The redistribution of replicate number 2 was similar to that of replicate 1 presented here. Here, to the right, we have a graph showing the breadth of coverage on the y-axis and the number of rates on the x-axis. For smaller genome within the Zymo community, like Staphylococcus and Lactobacillus, you can actually cover more than 90% of the entire genome, even at 10 million reads. And for the community members with larger genome, like Saccharomyces and Cryptococcus, you still notice an increased breadth of coverage that will definitely boost the accuracy of detection. So, we have seen how Jumpcode human DNA depletion kit is effective in depleting human genome without introducing any obvious bias, as can be seen with the Zymo microbial fold change. Now we want to look at how our Jumpcode host depletion kit performs compared to existing host depletion kit using actual clinical sample, which was saliva sample in this case. We conducted an experiment to check how our Jumpcode human DNA depletion kit compared to other host depletion kits out there. The initial competitive testing was between Jumpcode human DNA depletion kit and Mozyme Molysis Basic 5 kit. With the Mozyme Molysis Basic 5 kit, you have the option of using a DNA extraction kit of choice to extract the microbial DNA after depleting the human genome. To make sure that there was no bias introduced by DNA extraction method after the depletion of human genome from the saliva sample using Mozyme Basic 5, microbial DNA was extracted using QIAGEN blood and tissue total DNA extraction kit, which is the same DNA extraction kit that was used for the sample treated with Jumpcode Genomics human DNA depletion kit. The same lot of clinical sample, which was saliva in this case, were tested using the two host depletion methods. From the results, we noticed that Jumpcode human DNA depletion kit outperformed Mozyme Basic 5 because Jumpcode has the least impact on microbial composition. Jumpcode also has higher species count correlation between untreated and depleted samples, and Mozyme lysis method requires fresh sample. Here, we are looking at the correlation graphs generated using sequencing read data from the samples that were tested. On the y-axis is the reads per species in samples treated with either Jumpcode host depletion kit, which is to the left, the graph to the left, or Mozyme Molysis Basic 5, which is the graph to the right. The x-axis represents the reads per species in untreated samples. We notice a high level of correlation with the Jumpcode host depletion kit with an R-squared of 0.96. On the other hand, the Mozyme Molysis Basic 5 gave an R-squared of 0.86. This matrix further shows that Jumpcode human DNA depletion kit outperformed Mozyme Molysis Basic 5 kit. Now, let's take a look at how using Jumpcode human DNA depletion kit to deplete human genome in saliva and vaginal samples have increased detection sensitivity for low abundant microbial species. Here, to the left is bar graph showing read reallocation in untreated and Jumpcode human DNA depletion kit treated vaginal sample. For the bar graph to the left, the y-axis represents the number of reads allocated to each kingdom, while the x-axis represents the different samples. And to the right is a bar graph of fold change on the y-axis and the sample represented on the x-axis. Notice that in the untreated sample, on the bar graph to the left, 5.17% of the reads is allocated to microbial species, but with the vaginal sample in which human genome content was depleted using Jumpcode human DNA depletion kit, the reads reallocated to microbial species jumped 3.7-fold to 19.02%. And this increase as we will see next, resulted in detection of more microbial species in samples where human genome was depleted using Jumpcode Human DNA Depletion Kit. Here, to the left, we are looking at bar graphs with the number of bacterial species with read counts of 10 or more on the y-axis, while the x-axis represents different samples. Notice that in the untreated sample, on the bar graph to the left, there are 10 bacterial species with 10 or more reads, but with the vaginal sample in which human genome content was depleted, using Jumpcode Human DNA Depletion Kit, the number of bacterial species with 10 or more reads shoots up to 46, which is more than fourfold increase. And the very interesting thing is that all of the 10 species in the untreated samples are among the 46 species detected in the Jumpcode Human DNA Depleted Kit treated sample. This is illustrated in the Venn diagram to the right. Here, we are looking at results from saliva sample experiment, and to the left, we have bar graphs with the number of bacterial species having read counts of 10 or more on the y-axis, while the x-axis represents the different samples tested. Notice that in both sample number 1 and sample number 2, the untreated samples consistently have lower number of bacterial species when compared to the saliva samples in which human genome content was depleted using Jumpcode Human DNA Depletion Kit. And the interesting thing is that after depletion, for sample number 1, you detect 29 more bacterial species, while sample number 2, you detect 32 more bacterial species after depletion with Jumpcode Human DNA Depletion Kit. And for each sample, the bacterial species with 10 or more reads in the untreated sample are a subset of the species detected in the Jumpcode Human DNA Depletion Kit treated sample. This is illustrated in the Venn diagram to the right. So, with Jumpcode Human Depletion Kit, you can do more at lower cost, boosting your microbial read content to more than 3 times, resulting in the detection of more than 29 bacterial species, and all these benefits achieved in a one-day workflow. Additionally, with Jumpcode Human DNA Depletion Kit, you characterize your sample with confidence, because from all what we've seen, Jumpcode Human DNA Depletion Kit does not introduce any noticeable bias, and this applies to fresh and frozen samples. So, reach out to our sales team at sales@jumpcodegenomics.com and get your early access kit and begin to reap the benefits of Jumpcode Human DNA Depletion Kit. These benefits will include unbiased view of human microbiome because our method uses specific CRISPR-mediated depletion of the human DNA. You will also achieve improved microbial genome coverage previously obscured by human DNA. You will increase detection sensitivity of low abundant microbial species. And this is easily done in a single-day workflow and can be applied to extracted total DNA from microbiome sample, be it frozen or fresh samples. Now, before I end, I wanted to introduce you to another very exciting application of Jumpcode CRISPRclean technology, which we are working on. So, currently we have a CRISPRclean Plus product which performs well in removing human and bacterial ribosomal RNA from RNA-seq data. But in this experiment, we tested the depletion rate using the CRISPRclean Plus guide set, and on top of that, we added additional guide set designated as GS1 and GS2, and this amounts to approximately 1.3 million guides in the reaction. And after we performed this experiment, the depletion rate was phenomenal. In this experiment, we used contrived sample, which consists of 50% human total RNA, 50% Zymo Community Standard RNA, and we also spike in at 0.001% a SARS-CoV-2 RNA. In this experiment, there was one control where there was no Jumpcode CRISPRclean treatment, and there was also a sample set in which we only treat with the CRISPRclean plus guide set only. And the third set was the one that we treated with the CRISPRclean plus guide set, and also in addition to that, we added the GS1 and the GS2 guide set. In the end, we are looking out for how the coverage of SARS-CoV-2 genome, which was not targeted by any of the guide sets, will increase after the depletion of the human and the bacterial RNA. The y-axis on the bar graph is the depth of coverage of SARS-CoV-2, while the x-axis represents the different samples. Notice that the untreated control sample, SARS-CoV-2, has a depth of coverage of 0.6x, and with the sample treated using only the CRISPR-Clean guide set only, you notice that the depth of coverage is now 5.33x. And then, when you treat the sample with a cocktail of the CRISPR-Clean guide set alongside the GS1 and the GS2 guide set, the depth of coverage for SARS-CoV-2 jumps to a whopping 17.5x, which translates to a 26-fold increase. This was a very exciting outcome, and we will be building up on this to make sure that we have the tools to continue to fight the current pandemic while preparing for events of any new pandemic. Here is a graphical representation of the SARS-CoV-2 genome coverage. The y-axis is the depth of coverage, while the x-axis represents the 29 kb SARS-CoV-2 genome. The blue plot is the control, while the purple plot is that of the contrived sample that was treated with a cocktail of the CRISPR-Clean plus guide set plus the GS1 and the GS2 guide sets. So you can see why I'm excited about the future of CRISPR-Clean technology, and you have lots of options in our toolbox to enhance your research. I know this is a lot of information, but I'm definitely excited, and I will stop here so I can take your questions. Thank you.
Great. Well, thank you, Zenas, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, what steps were taken to ensure no microbial sequences were depleted?
Yeah, very good question. Thanks for that. Our Jumpcode genomic pipeline uses a very specific guide design pipeline in which the target that we were using in this case was ALU sequence, which is primate specific. And we also make sure that during the guide design, we filter it through pathogens website and also any other microbial website. And through all of those filtering, we, in the end, make sure that no other microbe is targeted. We are just like specifically targeting the human genome. So we did all the filtering through our specific pipeline to make sure that there is no off-target effect.
Great, thank you. I have another question for you here, and this one asks, will this protocol work with archived samples?
Yes, very good question as well, and I think that is where the strength of the Jumpcode host depletion product comes in, because most product out there that are doing this require that you have fresh samples. Jumpcode doesn't require your fresh sample, because we know it's difficult to actually get fresh samples. So with Jumpcode genomic host depletion protocol, you don't need fresh samples. You extract your sample, grab total DNA from your sample, and then you can be sure that jump code depletion protocol will only remove the human and then leave your microbial DNA intact.
Great. Next question here asks, the longest fragment lengths in my samples are about 10 kb. Do I still need to do size selection prior to your protocol?
Good question. So the current version of our product still requires, even though your sample is of 10 kb, we still require that you size select for the 15 kb plus, so that you will get the full benefit of the depletion. It's not that a 10 kb average size would not still deplete. You will still deplete, but you won't have as good depletion as you would with a 15 kb plus. So yes, we still do recommend at this stage that you size select for 15 kb plus.
Great. Got a few more questions in the queue here. I want to remind our audience that if they do have any more questions, please submit them now. We've still got some time here. So this next question asks, how is the depletion strategy different from other host depletion methods?
Yes, again, a very good question. We are very unique in what we do, and this is quite different. So now if you look at other methods, because human cells do have, doesn't have cell wall, but most microbial organisms do have cell wall. So other methods do capitalize on that, and then they will easily lyse the human cell with easy chemicals. And then when that is done, the DNA is exposed and then it's removed using enzymes. This process uses the microbial cells intact. Then you would remove that, remove the human cell, then go in and lyse the microbial cells, and that is how it works. But now we know that microbes like mycoplasma, they don't have cell wall, but they are also microbes. So those microbes will be removed. So we are different in the sense that you will not have to worry about if your microbe has cell wall or not. So you can grab everything as long as they are in there. In the end, you will only remove the human cell, the human genome from your sample. And there's also the problem of when you see, even though a microbe have cell wall, something like gram positive and gram negative, they have different levels of the cell wall. So gram negative will have a weaker cell wall than a gram positive one. So with that, you can also have a system in which those other methods would introduce bias, because they would also easily, when you restore your sample, those weaker cell walls will break open, and then you will remove your microbial DNA as well. So ours is very unique in that you don't need to worry yourself about your sample being fresh or you having microbial DNA in solution without having intact cells of your microbes. So we are unique in that we don't require fresh samples, and we also work with total DNA.
Wonderful, thank you. Next question here asks, does this depletion strategy bias toward gram-positive or gram-negative bacteria?
Very good question. So we do not observe any obvious bias. From the presentation, you saw that the Zymo microbial community that we were looking at have different GC content, have gram-positive and gram-negative microbes in there, and we did not see any bias in the boosting of each of those microbes. But we know that other methods do bias because they use selective lysis, which we know is not always perfect. So our method doesn't bias towards gram-positive bacteria.
Great. Well, thank you, Zenas. We're going to go ahead and wrap up here. Do you have any final comments for our audience?
Yes, so thanks, Kelly, for organizing this. It is quite exciting, and I would thank you guys so much for joining today. And again, if you have any questions, we are always ready to answer your question. And I think this is a very exciting product that you want to test and see it for yourself, how you would boost your discovery. And I would say again, thank you so much for your time.
Thank you. All right. Well, thank you one more time, Zenas, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Jumpcode Genomics, for underwriting today's educational webcast. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.